Good evening. Welcome to this problem solving session. My name is Nandika. I'm a PhD student at IIC Bangalore, and I'm going to be discussing content related to some content and some questions related to week 6 of the experimental biotechnology course, which was related to affinity chromatography. So, as always, if you have some doubts that you want to ask, you can always unmute yourself or ask it in the chat or anything. Meanwhile, I'll be discussing some problems, and feel free to stop me at any time. Just share my screen. So, over the past few weeks, we've been discussing a process called chromatography, which is basically a purification process that we use during protein extraction to ensure that we are able to purify our protein of interest. So, what that would look like in a protein purification workflow is something like this, where we are expressing our protein probably in a bacteria, maybe using yeast or mammalian system or some other system, but the principle will remain the same. Either you'll express it inside the cell or you'll add some secretory tag to it and it'll be sent outside the cell so you can get extracellular expression. And then you will recover a protein by cell lysis that will lyse the cell and allow the cytoplasmic contents to be released. After which we have to remove all the remaining cell membranes and so on through debris removal. And then once we recover the supernatant, or if you have extracellular expression, you're directly collecting the media, then we can proceed for purification. So what we're getting after all these steps is a very complex mixture of many proteins. All of them have different properties. And what we can do is take advantage of the different properties to selectively purify our protein of interest. So, as we've discussed in previous weeks, there are many different types of chromatography depending on which property of the protein you want to look at. So, one property that we discussed quite extensively was using the size of the protein in a process called size exclusion or gel filtration chromatography, where basically we have pores of varying sizes in the matrix, and because of that, different sized particles travel in a different way through the matrix, and thus we can separate them. But of course, there are many proteins which might have the same size also. So those can be separated in other ways. So, like we discussed, we can make use of the hydrophobicity of the protein or the charge properties of the protein. Or if we want to make use of some very specific interaction, it is called affinity chromatography. We're specifically making use of the fact that our protein will bind to some ligand. And that is a property of our protein and not of any other protein within our solubilization mixture. And so this can be a very specific way to isolate our protein of interest. So basically, our protein, which is this green thing here, is binding specifically to this triangular ligand here. And that's why it is getting stuck on the column. And all of these other proteins don't have that binding site. And hence they will just flow through this column without getting stuck on the matrix. Okay, that is the basic principle of affinity chromatography. So just in more detail, we already have, wait, I'll just, we already have in our stationary phase, we have our matrix with this ligand on it. And first we will equilibrate it with the buffer that we are using for purification. Then we will go for sample application. That means that we will put our mixture of proteins in the column, and wherever the complementary binding site is there, that protein will be able to bind to the column, right? Obviously, we have to do this in a condition that is favoring the binding. So maybe we won't use very extreme conditions at this stage. So we should choose some condition where our protein is stable and this binding interaction is strong. In case any of these other proteins are able to bind in some non-specific manner to the column, we want to remove those first. Now, obviously, because they're binding in a non-specific way, the strength of that interaction will be less than the strength of our regular affinity interaction, right? Because this is a very specific binding. And if this is just getting adsorbed onto the matrix, maybe that's not a specific interaction. So that can be eluted by washing the column with our buffer, right? After washing, we'll be left only with our bound protein, which we then want to elute. There are many different strategies for elution, which we'll discuss later, but mostly we can elute it either by changing the buffer composition or the buffer properties so that the binding is now destroyed, or we can use a competitive ligand. So it's called a counter ligand, where something that has a stronger binding affinity for this than our protein of interest. So that will cause our protein to be unbound from the column and it will come out as the elution. And then once you've eluted this, our column is ready to be used again. Okay. So if we look at a chromatogram of a regular affinity purification, it might look something like this, where we're just looking at how much protein is coming out at each stage. So first we will do the equilibration stage, where we're just passing our purification buffer. Then we will begin to add our sample, which contains many different kinds of proteins. Then as we pass more buffer, both the unbound material as well as any non-specifically bound material will come out of the column. Right? As we pass more and more buffer, the non-specifically bound protein will come out. Now we can change to the elution buffer. This is either going to have a different pH or it is going to contain some ligand that is going to compete with our protein. Once we change the elution buffer, now the binding between our protein and the matrix is destroyed, and so the protein will elute as a sharp peak, and now the column is free to use again. Okay? So this peak is what we're interested in this protocol. So like you can see, affinity chromatography can be a very specific way of separating our protein. So some interactions that we commonly make use of in affinity chromatography can be either natural affinities that exist already, that is called bioaffinity chromatography. So things like antibodies and antigens. We already know that these two will bind to each other. If an antibody is produced against an antigen, it will bind to it. So we can make use of this interaction in our chromatography itself, where you can immobilize either the antibody or the antigen in our column and then pass the other partner in the buffer, and that will bind to our column. Some other things like glutathione S transferase binds to glutathione. So we can use this to purify proteins which have a glutathione S transferase tag. If we're trying to purify some enzyme, then we can just immobilize a substrate or immobilize an inhibitor, right? An inhibitor would make more sense. You can immobilize an inhibitor on the column, and so the enzyme will bind to that. Okay. Another very commonly used chromatography is metal ions and histidine, which we will discuss. Okay. And just an example of what affinity chromatography might look like in a protein purification workflow. So here there's an E. coli cell extract where they're trying to purify a protein called manganese. Okay. And they're doing histidine nickel affinity chromatography, which as I said, we'll discuss later, where you can see that they have gotten a sharp peak after affinity. So this will contain manganese, but it might also contain some other contaminants. Okay? So this is what they're showing us. So this is the molecular marker. This is the material they were starting with. Okay? So this is what was being applied in the column. This is the flow through. So that means this is whatever, when we pass the sample, this was whatever that was not bound in the column. Okay? So none of this had any affinity for our column. This is early elution fraction. And this is the elution fraction. So here we can see that there are many kinds of proteins within this fraction. So although our protein will be one of these, this is not going to give us a very pure protein. So commonly what we can do after affinity chromatography is go for another form of chromatography, which is commonly size exclusion chromatography. Because here you can see many bands, but since only one of them is our protein of interest, the easiest way to separate them is by size. So here you can see after we perform size exclusion chromatography, We've gotten many peaks here, but this one is our protein of interest here, which we've gotten around 97 kilodalton. And you can see this protein is now highly enriched in the SEC fraction. So affinity chromatography can be a good intermediate step in the protein purification process, but you will often have to do some other form of purification after that. So just a regular overview. We have our cells with different proteins, but if you want to purify our protein of interest, either we should have some partner that binds to it, or we can add some tag to our protein. And that tag has some specific binding. So this could be like a histidine tag or a glutathione transferase tag. So we know that those will bind specifically to some ligand. So once we have either a tagged protein, we can then purify this through affinity chromatography and get rid of all the other proteins which are not tagged. Now, tagging a protein may not always be good for the protein. Okay, I mean, obviously it will help with affinity chromatography, but it comes with some disadvantages too. So, like we can see, when we have a tagged protein, we can purify it simply using affinity chromatography. Right? And it also has other advantages in that we can use the tag for other molecular biology purposes. So it can be very good for things like Western blotting and so on. You don't have to get an antibody against your protein, but if I know my protein has a His tag, I can just use an anti-His antibody. Right? Some tags can help with improving solubility and stability. Like there's a maltose binding protein, MBP tag, which often helps with solubility of our protein. Okay? It can also be used as a marker for expression, and also in structural biology, tags have a very different kind of purpose that's maybe not within the scope of this here. But of course, tags also have some disadvantages, that the tag may interfere with the protein structure and affect the folding and activity of the protein. So if the tag does not allow the protein to fold properly, then you can purify it, but you'll be purifying a non-functional protein. So this is something you have to keep in mind. Sometimes if we have a tag, we also want to remove it for further steps. So that will occur by cleavage between the tag and the protein. Uh, of course, you can only do this if your protein is stable for that period of time. And also the cleavage which you do will never be 100% cleaved. So you do have to keep this in mind if you're going to tag your protein. But I think the advantages outweigh the disadvantages, because if you have an untagged protein, yes, obviously you won't need to remove the tag, but purification can become extremely difficult in this case. So we'll just go through some quick questions based on this. So it's asking, affinity chromatography is based on dash. So the duration, flow through time, molecular weights, no. So these are all terms related to size exclusion chromatography. But affinity chromatography separation is based on specific interaction between the analyte and the ligand. Right? That is the definition of affinity chromatography. Affinity chromatography can be used for DASH, the study of enzyme substrate interactions. Possibly. I mean, you can have different affinity matrices with different substrates and you can see what kind of enzyme yield is there. But there are much more efficient ways to do the same experiment. So I would not go for this as the answer. Is it used for the purification of compounds from a complex matrix? Yes. The determination of protein molecular weights? No. You will not get any information about the protein molecular weight through affinity chromatography. The concentration of a compound? No. Because oftentimes you will elute in quite a large volume, so it's not effective for concentrating anything. The separation of proteins according to the isoelectric points. Again, this is not something that you will get any information about by affinity chromatography. So I would say the most appropriate answer here is this, but this you can also do to some aspect. Which of the following is not a highly specific biological interaction to be used in affinity chromatography? So, as we discussed, antigen antibody can be used, enzyme substrate can be used, receptor ligand can be used. Now, cation anion is not a highly specific interaction. This is just an electrostatic interaction. And it can take place between any pair of cation and anion. So, it is not a highly specific biological interaction that we can use for this purpose. Okay. So, coming back to chromatography, how can we make a column for the purpose that we want? Say I know I have a protein and I know it has a binding partner. There's some ligand I can use for affinity. Then how can I go about setting up this experiment? So the first thing you need to do is prepare the affinity chromatography medium. That is the stationary phase where our protein will bind. So here we can see that the column is made up of these beads, and the beads have multiple components. So first there's the matrix. Okay, this is the material of the bead itself. Okay, it should be chemically and physically inert because obviously it should not be affected by any of the buffers that we are using in our purification process. So even if you're using a buffer at very low pH, very high pH, lot of urea, lot of DDT, anything like that, it should not affect the column itself. So it should be chemically and physically inert. Then, of course, we have our ligand, which is where our target molecule is going to bind. But to attach the ligand to the matrix, there should be usually a spacer arm. Okay, so spacer arm will be conjugated to the matrix and then conjugated to the ligand, and this helps in effectively binding our protein of interest to the ligand, and I'll show you why. Okay, so first the matrix. It is an inert support onto which a ligand can be directly or indirectly coupled. There are some properties that the matrix should have so that it can be effective. It should not have adsorption, right? So our random protein should not be getting stuck in the column because that is defeating the purpose of using a specific interaction. If I'm getting non-specific interactions, then what is the point of using the column? Easily derivatized groups, because we want to attach our ligand easily. An open pore structure. So this is basically saying that if the bead is like this, then the only surface available for binding is this outside surface area of the bead. But instead, if I have a bead with pores, then I have actually increased the surface area on which my ligand can bind, right? So this bead will be able to have a higher binding capacity than this bead. So preferably we would want a matrix like this. And stability under a range of experimental conditions such as high and low pH, detergents, and dissociating reagents. Obviously, as we discussed, it should not be affected by the buffers that we are passing in our experiment. The ligand that we are using, that is the molecule that binds reversibly to a specific target, which enables the purification. There are two factors to select this ligand. It should have specific and reversible binding. Okay, in classical affinity chromatography, the binding between the ligand and our target should be reversible because this is our ligand and this is our target. We also want to be able to elute the target, right? So we can recover it. If it is irreversibly bound, then either we can't get the target out of the column or we will have to break this linker to get the target out. So now you cannot use this column again, right? There is a form of chromatography in which a covalent interaction is formed. It's called covalent affinity chromatography or immobilized affinity chromatography, which is a slightly different topic. But classically, the binding should be reversible between these two. Also, since we are going to use the ligand as part of our column, it should have chemically modifiable groups so that it can be attached to the matrix. And this group and this modifiable group should not be like near the binding site or anything, because if I have a molecule like this, okay, and my target is binding like this, and now I cannot bind it to the matrix like this, right? Because the binding site was here. Now I've destroyed the binding site, and so this column is not of any consequence for affinity chromatography. So it is important that we have some attachment to the column away from the binding site. Okay. Ideally, there should be very strong affinity between our this ligand and our target, but it shouldn't be so strong that we cannot elute our protein. So ideally, it should be between 10 raised to 4 to 10 raised to minus 8, the dissociation constant. If the dissociation constant is very high or very low, then perhaps you have to consider using harsher elution methods. Okay. Yes, so like I mentioned, when we attach the ligand to the column, often there will be a linker. And this is the reason why. So if the ligand is attached directly to the matrix, then you can see that it's not very easy for our target to bind, right? Because the binding site is all the way inside our target, but there's no space for a target to bind properly. So it's facing steric hindrance while binding here. So the easiest way to overcome this problem is just to add a spacer, which is giving this much gap, right? So our target protein can now bind in whatever way that it wants. And this will give a far more efficient binding. So here you can see in this, we're able to recover some protein, but it's a very small amount. Whereas once you have added the spacer, the amount of protein that we have recovered is much, much higher, right? So most groups will have a spacer arm between them and the matrix. Okay, now our target is bound to the matrix. Very good. Now we want to elute our target. How can we do that? Now there are many ways. The simplest case is that we will just change the binding affinity. So we can do that just by changing the pH, maybe. Right? I change the pH. Some of the groups, amino acids that were involved in binding have now gotten protonated or deprotonated or something. So now the interaction between the two is destroyed. So this is just the simplest case. A change of buffer composition elutes the bound substance without harming either it or the ligand. Right? This is very simple. So the column is also not harmed and we get our target protein. The other way is to use extreme pH or high concentration of chaotropic agents. So basically what this is going to do is denature your protein. Now once the protein is denatured, it will lose its tertiary structure. And once it has lost its tertiary structure, it will not be able to bind to your target anymore. It's most likely not going to be able to bind. Now, if your protein is very stable protein, then maybe you can refold it after this step, and then this is not a problem. But some of us don't work with stable proteins. Some of us work with fairly unstable proteins. And in that case, this is not a method that we will use because it can cause permanent damage to our protein. Now, the other way we can do is specific elution, right? So these are both non-specific elutions. Right? Anything that is bound to the column will be removed by these cases. But we can also do specific elution, and this is by adding a substance that competes for binding. Right? So here we can add either a substance which is competing for the ligand, right? So both this and a target have the same binding site, but this has a higher affinity for the ligand. So when we pass this, it will displace our target protein and it will bind to the ligand instead. The target protein will come out. The second option is that we can use some other ligand which has a higher affinity for this. So this is competing with our immobilized ligand. So this will bind to our target instead, and so it will come out of the column. Now, obviously the difference here is that here the target that you are eluting will have some bound ligand with it, whereas here it will be a free protein. So depending on what your application is, you can choose which method you want for elution. So like we mentioned, here are some non-specific methods for elution. One is by changing the ionic strength. So this you might remember from ion exchange chromatography, where we used a series of higher salt concentrations to elute our protein, right? Because we were just changing the ionic strength of the buffer that would change the specific interaction between the ligand and the target. So we can use, say, an NaCl gradient, and that will cause elution of the protein. The other option, of course, is pH elution, because that is going to change the ionization of the charge groups, and this can affect the binding sites. If now, if you are going to change the pH, it is better that when you're collecting the elution, you neutralize the solution itself. So, for example, if I'm going to elute my protein in acetic acid, then it is better that when I'm eluting the protein itself, I will add some buffer, or I will keep some pH of some basic solution in my elution tube, so that the solution will get neutralized there and then, and our protein will remain stable. Okay. Now, this was non-specific elution. If you want to do specific elution, you can do something called competitive elution, where you have selective eluents. This competes either for binding to the target protein or binding to the ligand, as we just discussed. For elution, it is common to use a concentration that is 10-fold higher than that of the ligand. Right? Obviously, if it is going to compete in the binding, it should have a much higher concentration present. Now, other elution methods, these are not commonly used, but it's good to keep in mind that we can have substances that reduce the polarity of the buffer. These can help elution without affecting the protein activity. So we can add some samples such as dioxane and ethylene glycol. Now, of course, if none of these are causing our protein to elute, we can alter the structure of the protein itself, right? So we can denature the protein and the protein will come out. So we would use things like guanidine hydrochloride or urea. So these are called chaotropic agents because they cause protein denaturation. So if you can refold your protein after this, then well and good. This is a solution that you can use. So let's go through some questions then. Requirements for affinity chromatography are dash. Specific and stable ligand. Yes, of course. It should be specific and it should be stable, right? It should not degrade over the course of our experiment. Then what is the use? Degradability of the analyte. This is not required. Ideally, nothing should be degrading in an experiment. Availability of a high molecular weight ligand. This is not required. We need a ligand, but there's no necessity for it to be of high molecular weight. So this is not a requirement. Possibility to attach the ligand to the stationary phase. Yes, of course. If the ligand can't be attached to the stationary phase, then we can't use it. Highly concentrated solution of the target analyte. This is also not necessary, actually. You can have a fairly dilute solution and you will still be able to capture the protein in it. Reversible bond between the ligand and target analyte. Yes. In normal affinity chromatography, we do require the bond to be reversible so that we can elute it. Ligand affinity to the sample matrix. This is not required per se. We don't need the ligand to have any specific affinity for the sample matrix. We just need it to be able to bind. So we will say yes for now because we do require it to bind to the sample matrix, but it's not in the same way as we think of affinity chromatography. We just need some complementary group to bind the ligand. Okay, which of these is not true when selecting a solid matrix or column during affinity chromatography? The matrix should interact weakly with enzymes. Yes, of course. The matrix should be based on inorganic compounds. This is not necessary. We can have matrix based on organic compounds also. Like Stepharos is cross-linked agarose. The matrix should exhibit good flow property. Yes. The matrix should be highly porous. Yes. So the only false statement here is B. In affinity chromatography, the matrix should be chemically and mechanically stable. Yes, obviously. So dash refers to eluting a compound by creating competition between the bound enzymes with a suitable complementary protein in solution. So when we're doing competitive elution, that is specifically going to elute all our bound molecules. So it is specific elution, right? Non-specific elution would be using other methods such as changing the pH, changing the ionic strength or something like that. Like this. Which among these is not a way of non-specific elution? Solvent change is non-specific. pH change is non-specific. Reversible denaturation is non-specific. But competition to suitable complementary protein is specific elution. So this is not a non-specific elution technique. Okay. So just an example of the kinds of ligands that we can put in our affinity column. So for example, this is something called a blue Sephiroth column, where we have immobilized a dye called Cibicron blue, this thing, F3GA. Okay? This actually binds to albumin. This binds via electrostatic and hydrophobic interactions, and can be also used to purify things like coagulation factors, lipoproteins, and interferon. Right? Because it's not a natural ligand of that protein. It's just by chance that albumin happens to bind to this because it has a complementary structure. But since it's not a natural ligand, it's not a very specific interaction only for albumin. Okay? So, for example, we have our blue dye, Cibicron blue, which is conjugated to our bead. And we can see that our sample is human serum. Obviously, it will contain human serum albumin. We bind it to our mixture, our matrix, and then we have to elute it. So here you can see elution has been done with 1.5 molar KCl. So this is non-specific elution. We're not going to compete it out. We're just going to change the ionic strength of the solution. And you can see as the amount of matrix increases, or rather the binding capacity of the column increases, we are able to purify a larger and larger amount of human serum albumin. Okay? As soon as we change the elution buffer, we get this peak which is corresponding to albumin. So again, as we discussed, if we have a protein which has a natural substrate, right, then we can use that as the ligand instead. So here we're looking at purification of NAD plus dependent dehydrogenases with other enzymes with affinity for NAD plus. Right? So if I know my protein is going to bind to this, then why not immobilize NAD plus on the column itself? Right? So this matrix is called 2' 5' ADP Sephiroth 4b, which is some trade name, good for them. And here, as we discussed, we have the linker. This is the compound itself. And we can use this to purify a protein. Here it says to elute this, we should use low concentration of the free cofactor. Right? So here we have to do a competitive elution, NAD plus or NADP plus. So that will compete with our ligand here for binding to our target protein, and our target protein will be eluted. Now, these are some examples of what the spacer arm and the group on the end of the spacer arm will look like. So the spacer arm should be activated at the end so that we can bind the ligand to it, right? It should be possible to add the ligand to the end of the spacer arm. So you can see that common spacer arms have a length of 10 atoms, 14, 11, 9, 12. This is just to give enough space between the ligand and our bead so that our target molecule can bind properly. And obviously we have different spacers for binding different kinds of groups. So if I want to bind a protein or a peptide or an amino acid to my column as the ligand, then I can use these. So I can have NHS groups, cyanogen bromide, and so on. If I want to bind a sugar, so I can bind the sugar through a hydroxyl group, an amino group, or carboxyl group, depending on what my sugar looks like. Right? If I want to add a polynucleotide, right? say, for example, I want to separate mRNA. This would be a very common example. I want to separate mRNA. I know my mRNA will have a poly A tail to the end of it. So, what if I just have a column in which I've added poly T oligonucleotide, right? My mRNA should be bound in that column. So, I can make a column like that by binding the amino group of the polynucleotide. In the case of this, apparently there's no need for a backbone because it's just going to be complementary, right? It's not going to bind like this, it's just going to be complementary. And amino group we can couple through cyanogen bromide. Okay. So a coupling reaction, as you can see, is not very complicated. We just have to activate the matrix and mix it with the ligand solution. And then when that is done, we just have to block any of the remaining groups where our ligand is not added, right? Because we don't want it to react with any of the proteins that we're going to add it later. After the coupling is done, so here for example, here's Cephalose with NHS, N-hydroxyl succinamide. Okay. So this is the linker with N-hydroxyl succinamide, this thing, which is the activated group on the end of the linker. And this is going to react with any amine group, primary amine group, within our ligand. So once this is done, our primary amine group has been attached over here, right? So our ligand is now attached to the column. After this reaction is done, we can wash the matrix with high and low pH to remove any excess ligand or any excess reaction byproducts. And now this is ready to use for affinity chromatography. Similarly, since we discussed NHS, another way we can use it is using cyanogen bromide. Okay. So cyanogen bromide, when we are adding it to Cephalose, it will react with the Cephalose, okay, and form an ester over here. So once we have activated the Cephalose, we can then add any primary amine, right? Anything with a primary amino group, and that will already react with this ester group to form a spontaneous bond. Okay, so this coupling occurs spontaneously. Okay, and in this way we can attach. Here you can see add cyanogen bromide, add a primary amine group, and we form this isourea linkage. Okay, where our ligand is now attached over here. So this gives us a lot of flexibility in what kinds of ligands we can attach, because this doesn't require any harsh conditions or anything. So this reaction takes place spontaneously. Obviously, one very obvious kind of specific interaction we can use for affinity chromatography is antibody antigen, right? So if I have a protein, I can generate an antibody against it. So I will use that as an immunogen, and I can say inject it into mice or into camels or something like that. And then once it is injected into the mouse will produce antibodies against this antigen, right? So I can then I can give a regime of several immunization doses so that these antibodies will be produced. Then I can isolate the spleen of the mouse, create a hybridoma, check, screen all the antibodies produced for ones which are binding to my protein of interest. And in that way, I can generate an antibody against the protein that I'm interested in purifying. So you can see a typical immunoglobulin antibody will look something like this, where you have two heavy chains, two light chains linked by disulfide bonds. As you are probably well aware, there are 5 major classes of immunoglobulins. We mainly use IgG immunoglobulins, but depending on your purpose, you might look into the others as well. These are all monomeric except for IgM, which is pentameric, and IgA, which can sometimes get a dimeric structure. But what you do also have to remember is that the paratope, or the part which is binding to our antigen, is only really within this region of the antibody. Okay, so for the binding purpose itself, there is no need to have this Fc region. So you can also have smaller versions of the antibody without the Fc region. So for example, without the Fc region, if you just link one set of heavy and light chains, okay, you can produce something called an scFv, which is still able to bind to your protein, but it's much smaller than the full antibody. Okay? And there are many variants on this. You can have a nanobody where you only have the heavy chain. You can have a megabody. There are many variants of antibodies you can use. But the point being, you can generate an antibody against your specific ligand. Now, since this is a protein, you can then couple it to your matrix through something like this. You can use either cyanogen bromide reaction or an NHS activated cephalose to couple this to our column. And now we can easily purify and specifically purify our protein of interest. Okay? There are also proteins which actually have affinity for antibodies. And this can be good if you want to purify antibodies for any other cause. So these are surface proteins found in the cell walls of bacteria. They're called protein A, protein G, and protein L. Okay? And these will specifically bind to immunoglobulins and are best for purifying IgGs. So protein A, you can see, has 5 binding sites, protein G has 3, protein L has 5, but protein A and G bind to the Fc region, that is, protein A and G bind to this region of the antibody, while protein L is binding to this region. Okay? So depending on what kind of antibody you want to purify, you can select one of these resins. So for example, in our lab, we purify an antibody using a protein L resin. And it's very convenient because we just add it to our cell lysate, we incubate it, we elute the protein non-specifically using acetic acid, and we immediately neutralize it using tris, and we get a very high yield of the protein. So it's a very effective means of purification. So just some more questions. So acid phosphatase is to mannose, as alpha amylase is to DASH. So mannose is a substrate of acid phosphatase that can be used in affinity purification. Similarly, for alpha amylase, the substrate of these that can be used is glycogen. Okay, glycogen is the substrate that can be used for affinity. Acetylcholine esterase can be purified using affinity chromatography with the use of DASH as a ligand. So here, of these options, acrinidium is the only ligand that can be used for acetylcholine esterase. Okay, these are just textbook questions based on pairs of affinity ligands. Okay. Affinity chromatography can be used to purify biomolecules using corresponding resins to which they specifically bind. Find the correct match. Okay. Carbohydrate resin and biotinylated biomolecules. No, this is not right. So a biotinylated biomolecule will not bind to a carbohydrate resin. Glutathione resin and protein hexahistidine tag. Again, this is not the correct binding pair. Nor is nickel NTA with protein with GST tag. So the only correct binding pair here, as we just discussed, is protein A or protein G resin with an immunoglobulin. Okay. Affinity chromatography is used to isolate biomolecules bound to one another and study their interaction using a ligand that specifically binds to one of the biomolecules in the complex. This technique is termed as DASH. Okay. So, is it SEC? No. Is it IMAC? So, IMAC is a very specific form of affinity chromatography. So, this is one of the right answers. Okay, this is called immobilized metal affinity chromatography, which we will discuss later. But what have they said? Bound to and study their interaction is blah blah blah. We can also use this to do a pull down assay. Okay, pull down assay just means that I am trying. You may also hear this term as immunoprecipitation, where I am purifying a complex, right? So basically, I'm using interaction between two proteins I'm going to isolate one of the proteins. I'm going to look at what its binding partner is. So this is also something you can do using affinity. To elute the desired analyte, it needs to be freed from the analyte ligand complex. Obviously, right? It is bound to our column, but we also need to elute it. This is achieved by adding in a competitor that binds more strongly than the ligand. This is competitive elution. This method is called specific elution. Non-specific elution is achieved using dash. pH change? Yes. Solvent change? Yes. Reversibly denaturing the analyte protein? Yes. So all of these are non-specific elution techniques. Okay, yeah, just give me a second. So like I mentioned, one of the most commonly used categories within affinity chromatography is called immobilized metal affinity chromatography, or IMAC. And this is specifically against a polyhistidine tag. So how this works is that you express your protein, and at either the N or the C terminus of the protein, you will then add a sequence that codes for many histidines in the row. Okay, so this will be commonly between 6 to 10 histidines. I think most common you will have 6 histag or an 8 histag. Right, so here this is a DNA sequence for a 6 histag, but you can have more. You can have up to 10. Okay, this is going to obviously going to add a little bit of size to your protein. It's going to be almost 840 Daltons. This actually, so what happens is this histidine can bind to nickel ions, or not just nickel, but a number of metal ions, as we'll see, and that can be used to purify this protein. Okay, elution conditions, how can we remove it from the column? One is using excess histidine. It's going to compete with a histidine tag. We can use a pH shift. So we are going to destroy the interaction between histidine and the metal. Or we can use imidazole, which is a small molecule with a structure similar to histidine. So it's acting again as a competitive elution. So this is what the structure of the complex with the nickel would look like. So the the matrix would generally look something like this. As we discussed, there's a bead, here's a spacer. This is something called NTA, trinitroacetic acid. Okay, this basically coordinates one nickel ion. Okay, nickel NTA is a chelator and Ni2 plus as the ion. So this is called nickel NTA. And what the nickel ion is then able to do is to bind to two histidines which are separated by this much distance. Okay, so basically our tag should have at least 3 histidines, so that these 2 histidines are in the correct place to bind to nickel ion. Okay? And just to make sure that this interaction takes place properly, we will normally have multiple histidines in the tag. This is a 6x hist tag. So it is not detecting one histidine, it is detecting 2 histidines separated by a specific distance. Okay? So that's why it is less likely. I mean, every protein in your mixture will probably have histidine, but not every protein will bind to the column for this reason. Okay, it's detecting a specific spatial arrangement of those 2 histidines. Now, often you'll hear it referred to as nickel NTA chromatography, but there are also other metal ions that can be used. Okay, so here you can see that we can also use copper, nickel, zinc, and cobalt, but there is a trade-off. Okay, so copper has very high affinity for the histag. It means a lot of protein will bind, but very low specificity. That means, okay, we'll get a lot of protein, but it will not be very pure elute. So then that is just increasing our work in the downstream processing stage. So it is better to maybe make a compromise. We should choose something that has a high affinity, but it should also have some decent amount of specificity. Okay, so that is why you commonly see nickel used, where it has slightly lower affinity than copper, yes, but it has a higher specificity. So you will get a more pure product. If you really, really want a pure product, you can use cobalt instead. Okay, this is a different kind of resin called a talon resin, which has extremely high specificity. So this will help you get rid of any contaminating proteins in your mixture, but you will get low affinity. So depending on your purpose. So for example, I work in a lab where we do cryo electron microscopy. And for that we need extremely pure protein. We should not be having other proteins there in our sample. We only want to study one protein. And so when we do affinity chromatography, we use a cobalt coupled resin because we want extremely pure protein. Okay? So like they mentioned, the chosen metal ion is always a trade-off between affinity and specificity. So how does the elution work? So you can see here we have a nickel ion which is binding to these two histidine rings, and it is being eluted by at the addition of a high amount of imidazole. So this will be something like 250 millimolar, 300 millimolar, 400 millimolar imidazole. And here you can see the structure of imidazole is the same as the structure of the histidine ring, right? So it is basically competing out the histidine. So here we're getting a very specific elution using imidazole. So most commonly you would get beads, you get the resin as beads which are stored in ethanol, and then you can use it using a gravity column like this. So you will take out some of the beads, okay, you'll put it into your gravity column and it will settle at the bottom over here. Then you can pass your purification buffer through this, and that will cause all the beads to settle properly at the bottom of the column. Then we can add a sample. As the sample is passing through the column, it will bind to the nickel NTA groups and it will be immobilized in the sample, and all the non-specific proteins will pass through. Then if you have any other non-specifically bound proteins, we can wash it. We can wash it with a low concentration of imidazole maybe. That will remove all the non-specifically bound proteins. Now you can see after washing that only our protein of interest is left in the column. So then we can elute that using a very high concentration of imidazole. So obviously most commonly you'll see this, a gravity column, but you can also use a packed column, a pre-packed column for affinity chromatography. All this nice blue resin is the nickel conjugated resin. It looks blue. And you can connect this to your purification unit the same as you would connect an SEC column or an ion exchange column, and use it for affinity chromatography. Like I mentioned, we can also use, that was a nickel resin. This was a cobalt resin. It's cobalt NTA. It's called talon. Okay, like we mentioned, this will have a much higher specificity than the nickel NTA resin, so we should get a cleaner protein. The process will remain the same, in which we have to equilibrate it, then we have to load the sample, then we'll wash it, and then we'll elute. This is a common protocol for all affinity chromatography, but this is just something with slightly higher specificity. So for example, if we look at this gel, what we see in different processes is, this is the flow through. Okay, so this is what we get when we are just loading the sample. Okay, so here you can see all these proteins which are not bound, but it seems like this is my protein of interest, and a lot of it has not bound either. Okay, because this resin has a low affinity, so it is not capturing all of the protein which is there in my sample. Okay, this is the wash fraction, right? This is this step, and you can see all of this was actually bound to the column. Okay, it has been removed in the wash. This is the elute. You can see the elute is not very clean. There are a lot of other proteins also in this process, but our protein of interest has been enriched a lot. Okay. So this much was affinity chromatography. On this ladder, this side of the ladder, I have then used this elution fraction to do size exclusion chromatography. Okay, and you'll see that in the fractions of the size exclusion chromatography, like over here, I have a fairly thin, fairly clean protein sample here after SEC, right? So all of these contaminants have been removed through SEC. As you can see, they're in these fractions. So I will just collect these fractions and I will get a fairly pure protein sample. Okay. Now, uh, when we reuse the column, sorry, when we elute the protein, now theoretically there is no more protein stuck in the column, but and it should be able to be used again. But if we want to reuse the nickel NT beads again and again, it is better to regenerate the resin. Okay, and what that basically means is that this is our nickel column. We can basically remove the nickel, okay, remove all of the bound compound, remove any protein which is stuck, remove everything, and then just regenerate it. So we will add fresh nickel. Okay. So what this regeneration process looks like is usually first we'll wash the column with urea or guanidinium hydrochloride just to remove any protein that might be stuck in the column. Okay. Then we'll wash it with EDTA. Right? EDTA is a chelating compound, right? It's a chelator. So that will chelate the nickel ions and those will be taken out of the column. So after this, actually the column will be white because all the nickel ions have been removed. Then we'll wash it with NaOH, wash it with distilled water, and then we'll regenerate the column using nickel chloride. Okay? So this is going to regenerate the column. It's going to add fresh nickel ions into the matrix, and then we will wash the column repeatedly to get rid of any excess nickel chloride which is unbound. Okay? Then normally we'll take out the resin, we'll store it in 20% ethanol so that it is preserved, it's a good preservative, and then we can use the beads again. Now, if there's some issues you're facing in your nickel NT chromatography, what could be the reasons? Here, there is low yield of the protein in the purified fractions. So what could this be? There's no protein in the first place. That means maybe a protein was not expressed in the first place. So if this is the case, then you should go back and make sure that your protein is being expressed properly. And you can do that by quite easily by Western blotting. Nickel ions are being stripped. Okay, that means that nickel ions are not staying in the matrix. They're coming out of the matrix. And so obviously they will not be able to effectively purify your protein. Elution conditions are too mild. That means our protein is still stuck in the column. It is not being competed out by our elution buffer. So then we should change the elution protocol. Either we can increase the imidazole concentration or we can look into something like changing the pH. Maybe the protein is precipitated. Then it's not going to come out of the column. So then you have to maybe try and use less protein, try and dilute the protein, and so on. Or you can change the NaCl concentration. Maybe there are some non-specific interactions taking place. So something is binding non-specifically in the column. Then we can add a little bit of a detergent to change this. Then, as I say, low yield of eluted protein while using highly unclarified sample. That means our protein sample itself was very less. If we had a very complex mixture, the protein itself was very less. So then it's true that maybe this will not be a very effective sample for affinity chromatography. So we should first try to clarify the sample a little bit. We should try maybe